Alright, so let's take a look at a dihybrid cross. And let's just do a different color ink. Okay, so let's just pick two random letters to use. So we're going to use A, and we're going to go ahead and use B. Okay, so in doing this dihybrid cross, we're going to look at what Mendel actually did with his, where he used two plants that were heterozygous for both of these traits. So both parents were capital A, capital B, little b. And yeah, both of them were. Okay? So now we can look at the combination of alleles. So you've got two traits, which means you're going to have 4 different combinations because each one has 2 possibilities. Right? So if you've done FOIL in math, it's going to be kind of similar to that. Okay? You start with your capital A, and then it can't be combined with a lowercase a, but it can be combined with a capital B or lowercase b. So there are 2 of your gametes, capital A, capital B, capital A, little b. Okay? Now we're going to go back and take a look at our lowercase a. Okay? Well, lowercase a cannot be combined with a capital A. Okay? Can be combined with an upper b and a lowercase b. So there are other 2 gametes. Okay? So now we have each of our alleles combined with the other alleles that it can be combined with. Now because both of our parents are the same, these are our 4 alleles for both parents, or 4 gametes for both parents. I don't know what that was. There we go. Okay, so you just write in both of your parents all the way. Alright, so now I'm going to go ahead and erase all of this on the sides so we can just look at our Punnett square here, our dihybrid cross. Alright, so again, you want to kind of start with your dominant alleles, and you really want to start with one allele at a time. So let's go ahead and figure out our A's. So we've got a capital A here, which will go all the way down this column, and we have a capital A here, which will also go all the way down this column. We have a capital A here, so go all the way across this row, and a capital A here to go all the way across this row. And then we can fill in the rest of these that don't have 2 with their little a's. So now we've taken care of all of our A alleles. So let's go ahead and do our capital B's. So capital B's go down this column here and down this column here. Now we've got a capital B here, so it goes across, and a capital B here that also goes across. Lowercase b here that we can take all the way across, and lowercase b here, and now we go down. So we've got the b's and here as well. Alright, so now we've filled out all of our Punnett square. Okay, so let's list out our different genotypes. Okay, so we have capital A, capital A, capital B, capital B, capital A, capital A, capital B, lowercase b, capital A, little b, and then we start going heterozygous on the A's, and list out the 3 different combinations of B's that go along with that. And now we go homozygous with our A's. Okay, so now let's go through and tally up the number of each genotype we have. So capital A, homozygous dominant in both A and B. We have one right here, and that is our only one. So we just have one. Now, homozygous dominant in A, heterozygous in B. So we have one here and one here. All right, so that's two. Now, homozygous dominant in A, homozygous recessive in B. We have one right there. So you'll start to notice a pattern as we go through this, just matching up each of our different genotypes here. They're usually in diagonals from each other. You'll also notice that the double heterozygous genotype is the most common. Okay, so we have one here and here. So that's two. One right here. One, two here, and one here. All right, so you notice your pattern. You've got 1, 2, 1, 2, 4, 2, 1, 2, 1. Okay, now let's take a look at our phenotypes. So we've got dominant A with dominant B. Just going to shorten it that way. We've got dominant A. Apparently I'm not going to shorten it that way. And recessive B. Then we have our recessive A with a dominant B. And our recessive, that's why that looks weird. Should have written that as a lowercase a. Recessive A and our recessive B. Okay? So these are our four different phenotypes that we can have. Now, if we look through, we don't necessarily have to look through our Punnett square at this point because it's still kind of a little confusing. We do have our tallies, though, on the side here. Okay, so we're looking for a dominant A and dominant B. Okay, what will give us a dominant phenotype? So we have right here, that'll give us dominant and dominant. This will also give us dominant and dominant, as will this and this one. Okay, so now we add up those totals. So 4 plus 2 plus 2 plus 1. Okay, so we have 9 dominant phenotypes. Now let's look at dominant A, recessive B. We have one here and one here. So that's a total of 3. Now recessive A, dominant B, here and here. That's another 3. And then recessive is just 1. So this is where you get that 9 to 3 to 3 to 1 phenotypic ratio. Okay, so we went through our genotypic ratio first, and then we went through our phenotypic. All right, so this is basically what Mendel did. Okay, but he used, in this case, seed, both seed form and seed color. So you can see really easily, especially the way that this Punnett square is done, that both the yellow and the round phenotypes are the dominants, okay? And then the green and the wrinkled are the recessive. And you can go through and pick out our dominant yellows here, okay? We also have a dominant yellow with a recessive wrinkled, and then we have our recessive, and then our recessive color with our dominant shape. Okay, so that one's really easy to go through and point out all of the different phenotypes and genotypes. Okay, but that's basically how you set up a dihybrid cross. Now, if you have the time, the inclination, the paper, the space, whatever, you can do triple or trihybrid crosses, you could do quadruple crosses. You know, I've only gone up to a triple. I did it for fun. Don't know that it was all that much fun, but I enjoyed it, so that's weird. But anyway, okay, moving right along. So heredity patterns can be calculated with probability. Again, as we've seen, it's a lot more easy than you might think. So probability is the likelihood that something will happen. It's not a guarantee that you're going to get it exactly like that. Okay, so if you have two heterozygous parents crossing, but they only form 3 children, or 3 offspring, okay, you're obviously not going to have 75% of them have the the dominant phenotype, because 75% of 3 is not a whole number, and you're not going to split an offspring in half, and yeah, okay? So it's just kind of a likelihood. Basically, every time there's an offspring made, okay, it's a flip of a coin, right? What likelihood, what the probability will be, like if it lands on that probability or not. So basically, whenever, say, 2 heterozygous parents cross, their offspring have a 1 in 4 chance of being homozygous recessive. Okay? They have a 1 in 2 chance of being heterozygous like their parents. Okay? And then a 1 in 4 chance of being homozygous dominant. That's all that means. So probability predicts an average number of occurrences, but not the exact number of occurrences. Okay? You can actually have 100% would be homozygous recessive out of a heterozygous cross. Okay, that is possible. It's not likely. Okay, in fact, it's pretty unlikely, but it is still possible. So probability is the number of ways a specific event can occur over the number of total possible outcomes, which is what we were looking at earlier. So we counted the number of boxes inside the Punnett square, and that was our total number of possible outcomes. And then we tallied up the different genotypes or phenotypes that would give a specific outcome that we're looking for. Okay? And so that's how we calculated our probability. Like I said, it's a lot easier to do it with a Punnett square than it is to just do the straight up math part of it. So probability applies to random events such as meiosis and fertilization. You can also use it for things like coin flips, all different kinds of things. And I say coin flips because guess what? Here we go. Looking at probability, easy way to kind of visualize it is flipping a coin. Okay, so this is basically, you're flipping 2 coins. They are identical coins, okay, or you're flipping 1 coin twice, whatever. So the coin has 2 possible outcomes, okay, heads or tails. But because you're flipping it twice, okay, you have 4 possible outcomes. You could get 2 heads, you could get a heads and a tails, or you could get 2 tails, okay. So that's all that this is showing. All right, this is very similar, obviously it is a Punnett square, but this is very similar to how the alleles kind of separate that we talked about just a little bit ago. All right, so when I talked about my meiosis, I said that the gametes that are produced are genetically unique from each other. Now, in order to do that, they have to actually undergo a weird little process that we're going to talk about now. So sexual reproduction creates unique combinations of genes. So they basically take the parent's genes, mix them up a little bit, and then split them in half. Okay, and that's how you get your gametes. So independent of assortment of chromosomes in meiosis takes place, which means that it really doesn't matter what combination of the genes of the chromosomes that end up in each gamete, as long as there's 1 of each chromosome. Random fertilization also takes place, so you never know which sperm is actually going to unite with the egg cell and create a zygote. So unique phenotypes may actually give a reproductive advantage to some organisms, okay, whether or not it makes them more attractive to a mate, okay, like some birds, some organisms other than birds too, have like this very specific dance that they have to go through. Okay, so there might be a phenotype that kind of helps the male bird perform that dance and that attracts the ladies, okay? It could be that there's this moth that's better able to blend in with its environment so it doesn't get eaten, okay? So it's not dead, so it can reproduce, okay? So that would be a reproductive advantage. And because it's a reproductive advantage, then that organism gets to pass that phenotype on to its offspring. You can also get disease resistance. It's something that we generally try to breed into things like plants. You also get cold resistance sometimes, things like that. And then increased survivability, which I talked about with the different colors. All right, so we're actually going to have to stop here and pick up in the next video.